This lecture covers Chapter 24 on the digestive system. We're going to start with just an overview of the processes of the digestive system. You see them listed here: ingestion, mechanical digestion, chemical digestion, secretion, absorption, and defecation. So ingestion is simply eating. It's the intake of food into the oral cavity or mouth, and then of course you're going to chew it and swallow it, so that gives you your mechanical digestion and propulsion. A lot of the motility of the digestive tract is due to visceral or smooth muscle, but what we're going to find is that it depends on where you are. So if it's smooth muscle, it's simply propelling the food or the remains of the food through the tract. You're going to get these either rhythmic cycles of activity, or you're going to get propulsion based on the introduction of food. Some of the visceral smooth muscle activity, particularly in the intestine, is rhythmic, and it's controlled by pace setter cells. And what happens, usually if you're sleeping, and particularly if you get to sleep at night, you get these house sweeping, house cleaning waves of contraction that just kind of propel everything through the intestine and dump it on into the large intestine. And so in this way, anything that you swallowed that was inedible, whether it was a rock or your mother's engagement ring or whatever, it's eventually going to find its way out. So this sweeping activity is through a process we call peristalsis. And peristalsis, whether it's part of the rhythmic activity or something that occurred simply because you introduced food into the tract, it's going to be waves of contraction. And we're going to see that you've got basically two kinds of muscle tissue: smooth muscle tissue, circularly arranged, and longitudinally arranged. And let's just look at the pictures. So here you see the longitudinal muscle is external to the circular muscle. Here we've introduced a bolus of food, a small ball of food, into, say, the esophagus. And what's going to happen is the circular layer constricts on the side towards the mouth, and it's going to squeeze that bolus of food towards the other end. At the same time, the longitudinal muscle is going to contract, and that widens the tract and gives that bolus a place to go. And so that contraction of circular muscle behind and longitudinal muscle ahead kind of forces that bolus of food forward. A different kind of smooth muscle contraction occurs in the small intestine. We have this activity we call segmentation, where it's just kind of churning. It's just squishing back and forth. It's not moving it forward, but it's mixing the contents. That's what segmentation does. Chemical digestion occurs when the food is actually broken down into smaller molecules. This is mostly going to be due to enzymes, but also the acid in the stomach can break things down. Secretion, of course, you're secreting digestive fluids, which includes not just the enzymes and acids, but buffers and salts. So we have to absorb it. After all, that's really what this is all about. We call it the digestive system, but you could just as easily call it the absorptive system, because it's the absorption that you really want. You've got to absorb the nutrients across the digestive epithelium, and we're going to follow those processes as well. And then finally, get rid of the wastes. And these wastes are not just the remains of the undigested or indigestible parts of the food. You've shed epithelial cells. You've secreted bile, part of which is a waste product. And you can also, if you have bleeding into the digestive system, you can have occult blood. Occult just means hidden. So an occult blood fecal test tells you whether you've had any bleeding into the tract. Okay, so we're going to focus on the parts of the digestive system. We'll be following these through. The digestive tract is that entire muscular tube all the way from the mouth to the anus. A lot of people use the term gastrointestinal tract as synonymous with alimentary canal, but technically the gastrointestinal tract begins at the stomach. So it includes the stomach and intestine, whereas the alimentary canal includes also the mouth, the pharynx, and the esophagus. Accessory organs are things that are not part of the tract. They might be inside the tract or be outside the tract, but they're certainly associated with and function with the tract. So the accessory organs are associated with the mouth. That includes teeth, tongue, and salivary glands. Or with the small intestine, that will be liver, gallbladder, and pancreas. So here you have a diagram showing you the parts of the tract and what each part does, and we'll be going through those. So we're going to focus next on the lining of the digestive tract. We're going to see that this lining consists of certain consistent layers that are, it's just a pattern throughout the tract. It makes sense that the lining of the tract is going to be protecting you from dangers either in your food or something else you swallowed with your food or your own teeth. So you've got corrosive effects, you've got mechanical stresses like abrasion, and of course bacteria that might have come in. So as we look at the histology of the digestive tract, we find this pattern of 4 major layers: the mucosa, the submucosa, the muscular layer, and the serosa. And what kind of lining that mucosa is, how thick or how complex the different layers are, depends on what tract you're in. And we're also going to see folds. This slide doesn't include, there are folds in the esophagus, and those folds simply allow the region of the digestive tract to stretch when you put food in it. So we'll be looking at those as we go. Here you're just seeing the basic pattern. This is a slide of the small intestine. We'll focus on the features unique to the small intestine later. Here, let's just focus on our 4 layers. The mucosa is the innermost layer. The submucosa is deep to that. Then there's a muscular layer, and then the serosa. And the serosa, once you get below the diaphragm where you're in the peritoneal cavity, that serosa is the same thing as the visceral peritoneum. All right, so let's look at the mucosa. Here we're going to see this is the innermost lining of the digestive tract. It has 3 parts: an epithelium, a lamina propria, and a muscularis mucosa. So the muscularis mucosa is smooth muscle. The lamina propria is a very thin, delicate layer of areolar, loose connective tissue. And then the epithelium, well, it depends on where you are. So the epithelium can be either simple or stratified. In the oral cavity, the pharynx, the esophagus, and anal canal, it's a stratified squamous epithelium. Remember, that's the thickest kind of epithelium. And that's where you're going to have the most worries with not just friction and abrasion, but also with bacterial contamination. Starting at the stomach and running through the intestine and almost all of the large intestine, it's going to be simple columnar epithelium because, remember, we're trying to absorb stuff. So simple columnar allows maximum absorption with a thick enough layer to maintain the integrity of the tract. We're also going to find endocrine cells. Enteroendocrine cells are cells that are part of the digestive epithelium that are actually secreting hormones, and we'll deal with those with each part as we get to it. The lamina propria is always the layer of areolar tissue underneath of the epithelium and any mucous membrane. Remember, our mucous membranes include the lining of the digestive tract, the respiratory tract, the urinary tract, and the reproductive tract. So all of the things that you need to include to support that epithelium, mostly including blood vessels, but also a lymphatic supply. Remember, our lymphatic supply is not just draining lymph, it's also on the lookout for bacteria, or it's going to absorb perhaps even toxins that you've taken in. The muscularis mucosa is just a thin area, a thin layer of smooth muscle that is deep to the lamina propria, and it's not the same as the major muscle tissue of the digestive tract. It's just going to contract to produce those folds and relax to allow expansion. So you have longitudinal folds in your esophagus, you have folds in your stomach, you have folds in your small intestine, and that folding is just of that innermost layer, and it's due to this muscularis mucosa. Deep to that is the submucosa. This is a denser, thicker connective tissue. It's an irregular, collagenous, dense connective tissue. It helps bind and support the mucosa. It's not only going to contain blood vessels and lymphatic vessels, but very often it contains glands that are secreting things into the tract. It's got its own nerve supply, the submucosal neural plexus, that innervates any nerve supply to the mucosa and submucosa, whether it's sensory neurons, parasympathetic fibers, or sympathetic fibers. The muscular layer is the separate, deeper muscle layer. It's usually arranged in two layers, as we discussed when we talked about peristalsis. The inner layer is circular. When it contracts, it constricts. The outer layer is longitudinal. When it constricts, it kind of widens the tract. I don't want to say it dilates because dilation just sounds so passive. It's literally forcing the tract to get wider. So it's mostly for moving stuff through by peristalsis. And like the submucosal layer, it has its own nervous system. It has a myenteric plexus, and it is mostly parasympathetic because remember, parasympathetic activates resting and digesting, but there's some sympathetic fibers as well. Okay, so that myenteric plexus is the entire network of the nerve supply of the muscular layer. And while it's mostly parasympathetic, there are plenty of other neurons involved. In fact, the enteric nervous system is considered to be almost completely separate from the central nervous system. It has its own interneurons, and it is very sophisticated. If you just took all the other tissues away, if you could digest away the epithelium and the connective tissue and the muscle tissue, you would see this fine network of neural fibers just outlining the entire stomach and intestine because there's that many nerve fibers involved. All right, our outermost layer, the serosa, is the serous membrane that covers the outside of the tract. It's going to be the visceral peritoneum below the diaphragm because you're in the peritoneal cavity. Above the diaphragm, and you're talking just there about the esophagus within the mediastinum of the thorax, we call that layer out there, it's a different layer. It's not a serosa, it's an adventitia. Adventitia is just a dense network of collagen fibers that just kind of surrounds and replaces it. You also have an adventitia around your major blood vessels. So adventitia is just kind of a common term for a dense layer of collagenous tissue surrounding a tube. All right, so here you can see, again, we're looking at a section of the intestine, but you can see the three parts of the mucosa, and you can see, focus in on the muscularis mucosa in that top box, and then drop your eyes down to find the circular muscle layer and the longitudinal muscle layer of the muscular layer, and recognize the difference between those two. We'll come back and look at this diagram again to pick out the specific parts associated with the small intestine, but you can see in the submucosa you've got some glands there, you've got major blood vessels there, and lymphatic supply. And then find your myenteric plexus, the nerve fibers, and the submucosal plexus. All right, so we're going to focus our attention on the peritoneum. The peritoneum is the membrane that lines the peritoneal cavity. You learned in A&P 1 it has a visceral layer covering the organs and a parietal layer covering the surface wall. And it is a mesothelial layer. All of the serous membranes are. And it secretes a fluid that we just call peritoneal fluid. It kind of separates all the organs in the peritoneal cavity and prevents them from rubbing against each other and prevents adhesions from occurring. So it's really an important structure. The peritoneal fluid is simply there to reduce friction. And while you may produce 7 liters a day, you're not supposed to have that much at any one time. It's just supposed to be enough to keep the surfaces slippery and wet. But we know of a disorder called ascites, which occurs when you can't hold fluid in the blood, and that fluid will build up in any space that permits expansion, and the abdominal cavity is a major one. Ascites is a major sign associated with liver disease. Okay, the mesenteries are extensions from the peritoneal membrane. Remember, the peritoneum is just the covering of the organs or the lining of the cavity, but there are these sheets of membrane that connect them to each other that arose from them, and they're called the mesenteries. Meso for mesothelium, and enter means intestine. We've already seen that word root several times. So there are very specific mesenteries that connect the organs to the body wall, carry the blood supply, carry the lymphatics and nerves, and kind of keep the organs attached without letting them get all tangled up. And they developed during the embryonic period as one started from the backside, one started from the front, and you don't need to remember which things come from which. But here you can see a diagram showing you the development of those mesenteries. On the right figure, you can see the developing liver is covered with visceral peritoneum and anchored by a ventral mesentery, and then the digestive tract is anchored by a dorsal mesentery. That's just interesting to see where they came from. So the lesser omentum connects the stomach and the first part of the small intestine to the liver. The falciform ligament connects the liver to the diaphragm and also helps divide the liver into its lobes. The greater omentum covers extensively the anterior surface of the guts. It folds down and folds back up and connects to the transverse intestine, or transverse colon, and to the greater curvature of the stomach. And that's your beer belly. That's where fat can accumulate as lipid reserves. Obviously, the amount of fat being stored there can vary from one person to the next. The mesentery proper is the term for the part of the mesentery that actually attaches to the small intestine. And it's a fan-shaped. If you pulled the intestine out, you could kind of fold it out like a Chinese fan. And it contains the blood vessels that you've already learned in lab. And, of course, the lymphatic structures, including intestinal lymph nodes and the nerve supply. Parts of the mesentery actually fuse to the abdominal wall, and so there are specific structures that are actually behind the peritoneal cavity. We call these retroperitoneal, and among these are the duodenum and the pancreas. Most of the pancreas is retroperitoneal. Most of the duodenum is retroperitoneal. And you might already know that the kidneys and the adrenal glands are also retroperitoneal. All right, our last part is the mesocolon. Clearly, this is the mesentery associated with the large intestine. And the mesocolon of the ascending colon and the descending colon and rectum fuse to the posterior body wall, and that makes these retroperitoneal. Ascending colon, descending colon, and rectum are retroperitoneal. But you have a transverse mesocolon, and you have a sigmoid mesocolon that actually do still provide you that fan-shaped mesentery-looking structure. All right, so this just shows with a lot of the structures cut out where the mesocolons are going to attach and the mesenteries are going to attach. And here is a sagittal view so that you can kind of see the attachment point. So let's start with a falciform ligament attaching the liver to the diaphragm up at the top. Below the liver, attaching it to the stomach, is the lesser omentum. Follow the lower edge of the stomach down, and you see that fold of the greater omentum. Find the transverse colon, and you can see the transverse mesocolon attaching it to the posterior body wall. You can see all of the little segments of the mesentery proper attaching to the different sections of cut small intestine, and then the sigmoid mesocolon down there at the bottom of the abdominal cavity. All right, so now we're going to turn our attention to the accessory organs very briefly. We're not going to say a lot about these now. We're going to do this as a sort of a section-by-section approach. So here you've just got a list of what those accessory organs are and what they're going to do. And again, we'll come back to those. So next we're going to turn our attention to regulation. And there are 3 different ways that we regulate the functions of the digestive system. Local factors are just secreted locally. They're secreted by nearby tissues, and they have a local effect. And then, of course, your basic mechanisms of the nervous system and the endocrine system. So our local factors include components in the food that you've eaten, the pH of it, how much you ate, the intestine or the chemical composition. And you've got sensory nerve endings in the intestine that can detect particularly fats or proteins, and they're going to stimulate the secretion of specific hormones that are going to regulate differently. If all you ate was dry crackers, you would not be secreting the same enzymes as you do when you eat food with a lot of fat or protein in it. Stretching of the intestinal wall, stretching of the stomach wall as well can cause localized contraction. Even stretching of the esophagus can cause peristaltic waves. And then local factors include things like prostaglandins and even histamine. Histamine is used in inflammation, and here it is in the digestive tract where it's going to cause an increase in blood flow and other chemicals, way more than we have time to cover. I keep finding new ones anyway. All right, so our neural mechanisms include mostly parasympathetic, but also these localized visceral motor neurons that are going to do some local control. We've got the myenteric plexus to control muscular contraction, and we have the submucosal plexus to control glandular secretion. We have two kinds of neural mechanisms, the short reflexes and the long reflexes. Short reflexes are part of what we call the enteric nervous system. They're just that local reflex arc that doesn't even go as far as the spinal cord, let alone the brain. Long reflexes are going to have those branches that go into the spinal cord and brain. They're mostly through the vagus nerve, and you've got some control that way in the central nervous system. So long reflexes allow activity in the stomach to control what's happening in the colon. Short reflexes are just going to be really, really localized. So if you stretch the esophagus, it's going to affect just the esophagus. Our hormonal mechanisms, we're going to find two kind of different varieties. You have enteroendocrine cells. Those are the hormone-secreting cells that are in the digestive tract itself. And they're going to produce some hormones that affect digestion and appetite and metabolism in ways that we are only now beginning to understand. But then some others are secreted by the digestive tract, and they have longer-term effects. They're going to affect secretion of some of your accessory organs. So we'll get to them. We'll see how they all work. So here's the diagram out of your book, looking just at the neural control mechanisms here. You have your long reflexes and short reflexes, and if it's hitting the myenteric plexus, it's going to cause peristalsis. If it hits the submucosal plexus, it's going to cause secretion. But you've got the long reflexes that go all the way to the spinal cord and brain. And here are the hormonal control mechanisms. The hormones are secreted by the enteroendocrine cells. Like all hormones, they have to get into the blood, even if they're having a local effect. And then on the far left, the local factors, the stimulus for digestion based on the chemicals or just the presence, the physical presence of food in your tract. Okay, so now we're ready to embark on a journey, starting with the mouth and moving to the pharynx, esophagus, and so forth, all the way to the end. And then the last thing we'll do is come back and talk about the chemical digestion separately. So oral cavity, you've got sensory awareness of that. So you've got appetite stimulation or suppression. If you smell chocolate chip cookies baking, that's going to stimulate appetite. If you smell a skunk, that's going to inhibit appetite. You have the sensory analysis of food in your mouth before you swallow it. That includes not just the basic taste of sweet and sour and salt and bitter and savory, but also what we call the mouthfeel. How creamy is that? Or how crunchy is it? Mechanical digestion through teeth, tongue, and the palate. Lubrication by mixing with saliva and mucus, and then a little bit of chemical digestion, as we'll see. So the lining of the mouth, we already know, is a stratified squamous epithelium, and it's not as thick as in your skin. It's non-keratinized, and it's on the inside of your cheeks, the inside of your lips, and the inferior surface of the tongue. The surface of the tongue is actually specialized for friction and gripping, but also it has, of course, the taste buds in it. The vascular mucosa under your tongue is very thin, and that's a place we put drugs to be absorbed. Common one is nitroglycerin tablets for people with chest pains. And the mucosa of the cheeks and lips, we're not that worried about those. The oral vestibule is simply the space in front of your teeth The oral cavity proper is behind your teeth. The gingivae are the gums, and we're not that worried about those. If you didn't know what the uvula was, it's that little thing that dangles in the back of your throat. It has very sensitive sensory nerve endings. It can trigger hiccuping, and it's obviously a sensory device to tell when food is entering the throat. The arches, palatoglossal arch and palatopharyngeal arches, are simply the sort of subdivisions, and we're not that worried about those either. So here's a diagram. If you didn't study this in lab, you've got the muscles labeled, you've got the tonsils labeled. Just kind of look through that and see if you can make sense of it, but I'm not testing you specifically over the diagram. So here's another one. The only thing I wanted to point out here is the frenulum of the tongue. So the frenulum anchors the tongue to the floor of the mouth, and sometimes kids are born with an excessively long frenulum, which prevents the tongue from freely moving in the mouth. And I know of one case personally and read of a case recently of a kid who had a long frenulum and he didn't talk. 2 years old or 5 years old and not talking yet. And when the condition was finally diagnosed and treated, it's just a simple, kind of snipping of that frenulum. Kids talking in complete sentences. So that really can happen. Notice there's also a frenulum associated with the lower and upper lips. It's just, the frenulum is just the anchor, that thin mucous membrane anchor holding those movable parts, anchoring them in place. All right, so the tongue. We're not going to worry that much about the sensory surface. You learned sensation last semester. And we're not going to learn the names of all the muscles of the tongue. We're going to learn that there are two kinds of tongue muscles. And there's a little bit of an enzyme secreted. Lingual, remember, refers to the tongue. So lingual lipase is an enzyme that can break down fat. The amount of fat it breaks down here is really tiny, and it's probably more associated with increasing your taste sensation than anything else. So the tongue has a body and a root. The body, of course, is the mobile anterior part. And you have two kinds of muscles, extrinsic and intrinsic. Extrinsic muscles are the ones that anchor the tongue out to the surrounding structures, mostly to the bone of the jaw or the hyoid bone. And intrinsic tongue muscles are the ones that let you stick out your tongue or roll your tongue or flatten your tongue. And these are obviously all involved in chewing and speaking, but intrinsic muscles are entirely contained within the tongue. All right, a little bit on teeth. I don't test you a lot over teeth. You need to know the four layers: the dentin, the pulp, the root, the cement, and the crown. So dentin and pulp go from root all the way to crown. The root, of course, is the part that anchors the tooth into the jaw, and the crown is the part that projects up. So looking at the diagram, you can see that enamel covers the crown. Dentin and pulp cavity go all the way from crown to root, but the covering within the root isn't enamel, it's cement. And that cement attaches to the periodontal ligament, which holds the tooth in place. All right, salivary glands. You've got three major pairs of glands, and then you've got a lot of little scattered glands. The three major pairs are the parotid glands, the sublingual glands, and the submandibular glands. And they all produce saliva, but slightly different properties to each one. The parotid glands are the big glands that are just in front of your ears. These are the ones that when someone has mumps tend to get swollen. You probably won't ever see a case of mumps because we vaccinate against it. But the parotid glands have these big parotid ducts that empty into the vestibule right at your second upper molar, and sometimes you can actually feel saliva coming out there. The main product of the parotid glands is the enzyme salivary amylase, which breaks down starches. The sublingual glands obviously are below the tongue, and most of what they produce is a thicker mucus. The mucus not only lubricates, it also kind of glues the food together. And then you've got a bunch of little sublingual ducts all along the lingual frenulum. And then third, the submandibular glands are obviously associated with the mandible. They're interior to the mandible. And they do secrete some salivary amylase and some mucin, but it's more of a watery secretion than what the sublinguals do. All right, so there's your diagram showing all those glands. Saliva, you produce about 1 liter of saliva a day. Obviously, submandibulars are the most important, even though the parotids are secreting a lot of the enzyme. And other things you secrete, electrolytes and buffers and glycoproteins. Glycoproteins, remember, are just kind of gluey proteins that help contribute to the mucus. The antibodies you secrete are the A antibodies, the antibody type A. And antibody type A can actually protect you from bacterial secretions, bacteria in the food that you eat. You also secrete an enzyme called lysozyme, and lysozyme can kill bacteria, which is kind of nice to have. You'll find antibody A and lysozyme also in lacrimal secretions. So we're going to lubricate, we're going to moisten the food, we're going to bind the food together, we're going to keep the pH of the mouth right. We're going to actually wash the oral surfaces. This is why chewing gum after a meal helps to reduce your risk of plaque buildup in cavity. And the salivary amylase is the only enzyme of any importance. And remember, it breaks down starches. So regulation of the salivary glands is through nuclei in the medulla oblongata. You have a superior one and an inferior one. You don't need to worry too much about exactly where they are or exactly what they do, other than to know that they're parasympathetically stimulated. So when you think about eating and your mouth waters, or when you actually put something in your mouth, even if it's not food, the act of having something physically in your mouth or chewing can stimulate secretion of the salivary glands. So that's parasympathetic. The sympathetic is going to be more of an inhibitory. So if something is a turnoff, it's not palatable, it doesn't smell good, then salivary innervation can be inhibited by sympathetic stimulation. And this is why stress gives people a dry mouth. So you're just not secreting as much. Okay, mastication is our word for chewing. And clearly you're going to use the muscles of mastication, some of which you learned last semester. And you're going to use the tongue to manipulate that bolus of food, mix it with the saliva, and also to help get it swallowed. So as we move into the pharynx, you have three regions, naso, oro, and laryngopharynx. And only the oropharynx and laryngopharynx are supposed to have food in them. The nasopharynx should only have air. And then we move into the esophagus, which is at its upper end skeletal muscle, and at its lower end smooth muscle. Most of it's actually smooth muscle. And it enters the abdominopelvic cavity through an opening in the diaphragm called the esophageal hiatus. And in some cases, if the stomach pushes up through that hiatus into the thoracic cavity, that's called a hiatal hernia. It's not the kind of hernia you usually think about, but it can occur, and it can obviously impact your ability to have food move through the tract. So again, we're going to have parasympathetic and sympathetic fibers, but we also have local control of that smooth muscle through peristalsis. So we have resting muscle tone in the circular layer. This is a functional sphincter. That is, we at the top have an upper esophageal sphincter that's tonically closed. It only relaxes when you swallow. And that prevents air from getting down into the esophagus. At the lower end, the lower esophageal sphincter, also called the gastroesophageal sphincter or cardiac sphincter, is tonically closed. It only opens when food is going down through peristalsis. So it opens just long enough to let that bolus of food or that slug of liquid drop through, and then it closes again. And that prevents backflow of materials from the stomach. Returning to our histology, we have mucosa, submucosa, and muscular layer. Remember, there's no serous membrane above the diaphragm. We have an adventitia. Our epithelium is a non-keratinized stratified squamous epithelium. We have these large longitudinal folds that help the esophagus expand when you swallow. And those folds are maintained by the muscularis mucosae. You have esophageal glands. That shouldn't surprise anybody. They're going to be in the submucosa, and they produce mucus to help lubricate and protect the surface of the esophagus. We have our inner and outer layers of muscle for peristalsis, and then we have an adventitia on the outermost side. So here's a transverse section through the empty esophagus. And you can see the lumen itself, the white area in the middle, is very small, and you can see how folded that esophagus actually is. And here's a close-up of the surface. You can see the stratified squamous epithelium, the lamina propria, and in the muscularis mucosa, you've just got to look for those darker pink bundles of muscle fibers. All right, so deglutition is our term for swallowing, and the swallowing reflex actually is controlled by a center in the medulla called the swallowing center. You've got tactile receptors in the palate and the uvula, which can be stimulated by the presence of a bolus or by the presence of the accumulation of fluid in your mouth. Saliva in your mouth will make you swallow. And there are 3 stages. The buccal stage is obviously in the mouth. Buccal refers to cheeks. The pharyngeal stage and the esophageal stage. So in that buccal stage or oral phase, you're going to use the tongue to push the bolus of food against the soft palate and the back of the oropharynx. So it's mostly the tongue. Obviously, your cheeks can also contract, but mostly it's the action of the tongue. And this obviously can be voluntary or it could be an automatic reflex. Once that bolus moves into the esophagus, notice the epiglottis has folded down. The larynx actually rises up in the pharyngeal phase. And this is all under involuntary control. It's still skeletal muscle, though, and that's important because when someone has a stroke, to the part of the motor cortex that controls the muscles here, that impedes their ability to swallow normally, which, first of all, is very frustrating, and second of all, causes coughing fits if they get food or liquid trickling down into the larynx and down into the trachea. And then our third phase, the esophageal phase, the smooth muscle of the esophagus takes over and you have peristalsis all the way down, and at the very end you relax that lower esophageal sphincter and let that food flow right on into the stomach. So the relaxation of the lower esophageal sphincter is the end of deglutition. All right, so now we're in the stomach, and what we're going to do here is store food, mechanically mix food with gastric secretions, and a little bit of chemical digestion. How much chemical digestion occurs actually depends on what's in the food that you ate, but also how long it stays there. So if you eat a huge meal, a lot of the food kind of gets stacked up into the fundus, which is the top of the stomach, and that's where most of the storage is going to occur. In the main portion or body of the stomach, you've got lots of muscular contractions there, and they're going to mix. Each bolus of food is going to get liquefied into chyme, and we're going to call it chyme from here on until we get to the end of the small intestine. So the stomach is shaped like sort of J-shaped. It has a lesser curvature where it attaches to the lesser omentum, which attaches to the liver, and a greater curvature on its lateral side. The greater curvature is attached to the greater omentum. And, of course, lots of variability based on you and what's in there. But as we look at the regions, the cardia is just the region closest to the heart. It's really not related to the heart, but it's just that little region around where the lower esophageal sphincter is located, and sometimes that is called the cardiac sphincter because of that. The fundus, the body, and the pylorus we'll see in a diagram in just a minute. So the cardia is kind of like an extension of the esophagus. You got mucus glands to help lubricate. The fundus is kind of quiescent. It's not that active in secreting or in contracting. So the big mixing tank is the body. And then the pylorus, the part at the end, is that skinny region between the body and the duodenum. And it's going to have peristaltic waves, which are going to pass food through to the next region. Guarding the exit from the stomach is the pyloric sphincter, and this helps prevent leakage of acid-rich stomach secretions into the duodenum, but it also prevents backflow. The pyloric sphincter has a kind of a twitchy trigger. If anything solid hits it, it tends to clamp shut, and that keeps all solids in the stomach until they've been thoroughly liquefied. So here you can see a cadaveric view showing you the position of the stomach within the abdominal cavity. And here you see the diagrammatic view with a fundus at the top, that rounded portion above where the esophagus connects. The main portion, the body, is the biggest part. You can also see internally the folds in the stomach. These folds are called rugae. And you've got an extra layer of muscle here in the body of the stomach. You have a longitudinal layer and a circular layer, but you also have an oblique layer, which increases the ability of the stomach to contract. So rugae are the folds, just like the esophagus has folds. The rugae fold to allow, it increases the surface area so that it allows for stretching to occur. And our extra muscle layer is called the oblique layer. And again, it's mostly in the body of the stomach. Histologically, if you look at the lining of the mucosa, we already knew it was a simple columnar epithelium. And it is thrown into a series of folds. These deep, deep folds form gastric pits, which are just the openings of gastric glands. And the gastric glands are the secretory cells of the stomach. The gastric glands have 3 kinds of cells. 2 of them secrete, and those are called parietal cells and chief cells, and they secrete the gastric juice. So here in the diagram you can see the gastric pit, which is the opening to the surface of the stomach. These have a lot of goblet cells to protect the stomach with mucus secretion. And then we'll look at the next slide to show an up-close view of the gastric glands. Here you see parietal cells and chief cells, but you also see G cells. G cells are the enteroendocrine cells. They secrete a hormone called gastrin because it comes from the stomach. Parietal cells secrete a couple of things. They secrete hydrochloric acid, and a factor to protect vitamin B12 called intrinsic factor. And then the chief cells secrete the enzymes of the gastric secretions. So parietal cells produce intrinsic factor, which is something that actually binds to vitamin B12, prevents it from being absorbed or destroyed by acid in the stomach, and carries it all the way through until it can be absorbed in the large intestine, actually. And then parietal cells also secrete hydrochloric acid. The process by which they do that is really fascinating, but we're just not going to get into it right this second. Chief cells secrete any enzymes. The main one in an adult stomach is called pepsin. Pepsin's a proteolytic enzyme, which means it digests protein. So to protect your own cells from being digested, it's secreted inactively as a precursor called pepsinogen. And when it mixes with the hydrochloric acid, when they're secreted together, then that activates it to the pepsin, and pepsin can break down proteins in your stomach. All right, so now we're going to look at this really cool process by which hydrochloric acid is secreted. We're going to see, actually we've already seen this before. We saw this in the transport of carbon dioxide in the respiratory system, and we'll see it again in the pancreas. So we have this enzyme, carbonic anhydrase, which I hope you remember from transport of CO2 by the blood. And so we're going to take CO2 and water in the presence of carbonic anhydrase and turn it into carbonic acid, H2CO3, which immediately dissociates into bicarbonate and hydrogen. The key to secretion is which way do these things go? In your red blood cells, the hydrogens were buffered by hemoglobin. The bicarbonates went out to the plasma. Here in the stomach, the hydrogen is actively transported out into the lumen. Going along with it passively is chloride, and there's your hydrochloric acid. The bicarbonate that is produced is actively exchanged for chloride and goes into the bloodstream. And because it's a base, it's going to raise the pH. We call this the alkaline tide. We'll see when we get to the blood flow that it's going to get canceled out once the blood from the pancreas joins in. In newborns, there are a couple of other enzymes that are secreted, and these are associated specifically with milk being the main food for a newborn. Renin, notice renin has 2 N's. We're going to meet another thing called renin or renin, R-E-N-I-N, so let's keep them straight. Renin coagulates milk. Gastric lipase breaks down milk fat, starts the digestion of the lipids in the milk. The main job of renin is to just simply coagulate the milk. and make it solid. So it takes all of the milk solids and just kind of coagulates them all. So if you've ever seen baby puke, baby vomit, it's like curds and whey. It's like cottage cheese. And that's because all the milk solids have been coagulated into these little clumps. That holds the food in the baby's stomach longer, which lets both you and the baby sleep longer. And in fact, rennet is used commercially to make cheese. So that's exactly how cottage cheese is made. You mix milk with rennet and you get this coagulation, and they drain off most of the fluid. Okay. The glands in the stomach, the pyloric glands, are you're not doing any more active enzymatic secretion there, so all your gastric glands are up in the body of the stomach. And in the pyloric region, you're back to mucus secretions like you were up in the cardiac region. And then our G cells, back to our G cells. The G cells produce our hormones. We are not going to learn all the hormones. We simply can't keep up with them all. So the one I'm going to ask you to remember is gastrin. Gastrin stimulates the stomach activity. Okay, so chemical digestion in the stomach is mostly by the action of the hydrochloric acid. Acid really does break stuff down. It can completely dissolve metals. It can help start the digestion of proteins, and it helps to liquefy everything. You don't absorb a lot of nutrients in the stomach. Two things that I know of that can be absorbed in the stomach are alcohol and aspirin. They are either unaffected by the pH or helped by the low pH. So regulation of gastric activity, there are 3 levels of all one we call your head phase, the cephalic phase, the gastric phase, the stomach itself, and then the intestinal phase. And all 3 of these can be either inhibitory or stimulatory. So the cephalic phase occurs when you have just the thoughts of food or the smell or sight or taste of food. Those are all positive reinforcements. If it's something disgusting, obviously that would be inhibitory. And for the cephalic phase, you're going to use the vagus nerves. You remember that these are the main parasympathetic nerves, and parasympathetic stimulates the digestive tract. So the cephalic phase is positive, happy thoughts of eating. And it could also be, the presence of food in your mouth is all going to stimulate. Just kind of get your stomach ready to go. So it's going to stimulate secretion of gastrin, and it's going to stimulate the secretory cells, and it's going to stimulate contraction, which is going to make your stomach gurgle. So when we get into the gastric phase, it's actually the presence of stuff in the stomach. Even if it's not food, if you distend or stretch the stomach, those stretch receptors can stimulate stomach activity. But obviously, if it's food, you've got chemoreceptors that are going to be activated as well. And again, we're going to stimulate both the secretion and the muscle activity and the secretion of gastrin. And then in our intestinal phase, things get a little bit more complicated because the intestine has to let the stomach know when it is ready for more stuff to come through or when it's not ready. So you've got both positive and negative feedback here. When you've just put some food into the duodenum, peristalsis of pyloric muscle and relaxation of the pyloric sphincter puts just a little squirt of this acidic chyme into the duodenum, and the duodenum has stretch receptors and chemoreceptors, and those then can stimulate both neural inhibition, which is going to tell the stomach just hold off, and hormonal regulation as well. So let's look at the hormones being secreted. We have 3 listed down the left side: CCK, GIP, and secretin. And all 3 of these hormones are going to get through they have to get through the circulation. Remember, that's how hormones get there. So it may take as much as a minute for them to get there, but notice they all have inhibitory effects. So the presence of lipids, carbohydrates, proteins, whatever, low pH in the duodenum inhibits stomach activity, both neurally and hormonally. So we're going to turn our attention to the accessory organs, and then we'll come back and look at what those hormones are doing to the pancreas, the liver, and the gallbladder. So the pancreas, extends from the duodenum toward the spleen. Yes, it's retroperitoneal, except it's very tail. And it has three basic parts: a head, a body, and a tail. It has one main duct. A lot of people have an accessory duct. In fact, the pancreas starts out in the embryo as two separate parts. You have a ventral pancreas and a dorsal pancreas. And in most people, they fuse together into one. But in some people, they just keep a little separate tiny pancreas. And histologically, the pancreas is composed of digestive exocrine glands, but also endocrine glands. So let's get our endocrine glands out of the picture quickly. The pancreatic islets are the endocrine function, and they are just kind of scattered throughout. And remember that these are the ones that secrete insulin and glucagon, and we'll come back and talk about them separately when we need them. But for now, we need to talk about the acinar glands, the acini, and these are exocrine tissues. They are just little blobs of glands with ducts, and all their little ducts are interconnected to get to that main pancreatic duct. So here you can see the main pancreatic duct. In the head of the pancreas, you can see the little accessory pancreatic duct there. Each has its own connection into the duodenum. The one for the main pancreatic duct is called the duodenal papilla. Remember, a papilla is just a little bump. Internally, you can see that the bile duct joins with the pancreatic duct. And where they join together, they form a little structure that carries both bile and pancreatic juice called the hepatopancreatic ampulla. All right, so here we have our close-up of the acinar tissue and the islets. So you can distinguish them. The acinar tissue, each one of those has to open to a duct, even if they don't show them each opening to the ducts. And here you can see a histological view of the same thing. All right, so our pancreatic islets secrete insulin and glucagon, and there's some other hormones, and we'll worry about them later. The exocrine cells are the acinar cells, or the acinar glands, and they're the ones that secrete pancreatic juice. We've got enzymes, we have water, and we have ions, predominantly bicarbonate, which, remember, is going to raise the pH, and they're all controlled by hormones. The enzymes of the pancreas are an amylase, which breaks down starch and glycogen. We've already seen an amylase from the saliva, the salivary glands. The lipase is going to break down the triglycerides, nuclease is to break down nucleic acids, and a whole host of proteolytic enzymes. The proteolytic enzymes are like pepsin in the stomach. They have to be secreted as proenzymes, or they would digest their own cells. So we start with trypsinogen. This is the primary one, and we'll see that it's going to be activated in the duodenum by an enzyme secreted only in the duodenum. And then trypsin can activate the other kinds, including most prominently chymotrypsin and carboxypeptidase. All right, our liver. We're going to look a lot at the liver. It's a chief metabolic organ, so we'll come back and talk a lot about the metabolism of the liver in the next chapter. And its location, I'm sure you're already familiar with. It's divided into lobes. It's anchored by a falciform ligament superiorly, and there's also a round ligament anteriorly. The porta hepatis is the point of entry of the hepatic portal vein and the hepatic artery into the liver, and the blood supply to the liver is almost entirely through the hepatic artery. The hepatic portal vein is draining blood from the digestive block of organs. The cells of the liver are mostly hepatocytes. All hepatocytes are practically identical. They're a glandular epithelium. They look like little cuboidal epithelial cells. And all hepatocytes can do the same function. Depending on where they sit in the liver, some do more and some do less. But all hepatocytes are basically equal. So here is a transverse section showing you the liver and its connection through the lesser omentum to the stomach. And here you can see the main two lobes, the right lobe and the round lobe, along with the ligaments and the position of the gallbladder. And here you can see the porta hepatis with the main bile duct, the hepatic portal vein, and the hepatic artery. Notice that the inferior vena cava just runs along the posterior surface of the liver. We're looking below the liver with a backside being at the top of the diagram. And you can see the left hepatic vein labeled. The left hepatic vein is one of the three veins that drains the liver. There's also a right and a central hepatic vein, but they're actually buried into the liver. All right, histologically, the liver is divided into lobules. We'll look at a picture of those in just a second. Remember, the hepatocytes are the main cells, but there are also lots of macrophages in the liver. The old name for these is Kupffer cells. Calling them stellate macrophages is fine, but a lot of people just call them liver macrophages. The macrophages of the liver actually sit in the sinusoids, which are the blood vessels of the liver. Here you can see a couple of lobules. Let's look at the fluids being drained or supplied. In red, you have a branch of the hepatic artery. In blue, a branch of the hepatic vein. In green, you have a bile duct. So starting at a portal triad, let's look at the lobule on the right, move towards the middle, and there you see what we call a central vein, and that's where the blood gets drained. So between the hepatic portal branch at the portal triad and the central vein, you've got running in, mostly blue in the figure, all of those liver sinusoids, and those liver sinusoids are where the macrophages hang out. In tan, all of the little cells you see here are the hepatocytes. So in this close-up, you can see the hepatocytes are little tan cells. You can see the sinusoids, and inside of the sinusoids, you see the little pink stellate macrophages. Notice that the blood from the hepatic artery branch and the blood from the branch of the hepatic portal vein are going to merge together so that the oxygen supply and the supply of nutrient-rich food or nutrient-rich blood coming from the guts is just going to mix together. The cells that are closest to the outside, the outer edge of the lobule, get the best blood supply, they get the most food, they're the most metabolically active. The ones closest to the central vein are less likely to be involved in processing food. They're more likely to be doing the other metabolic processes of the liver. Okay, so there is the portal triad. And the hepatic portal system, we know, drains blood from the stomach, pancreas, spleen, small intestine, and large intestine. All right, so the bile system, the liver, it says the liver secretes bile. It's actually those hepatocytes that are forming the bile, and that bile just gets continuously produced and dumps into the bile canaliculi between the adjacent liver cells. These eventually drain into the hepatic portal triad, and eventually those little bile ducts coalesce into the right and left hepatic ducts, and they unite to form a common duct, which then joins with the cystic duct, which leads to the gallbladder, and that common hepatic duct and cystic duct form the common bile duct, and that's what dumps in with the pancreatic duct at the duodenal papilla. The duodenal ampulla. So, that was a lot. So, looking at the diagram here, you can see the gallbladder, the cystic duct, joining with the common hepatic duct to form the common bile duct. If you're not eating food right now, then all that bile being secreted by your liver is going to back up through the cystic duct backwards into the gallbladder, and the gallbladder is going to store the bile, and it's going to concentrate the bile by removing some of the water in the bile, and sometimes that can contribute to the formation of bile stones. Why do they color them green? Because one of the components of bile is the bile pigment, bilirubin, which is simply a waste product from hemoglobin. When your red blood cells get worn out, you dump the hemoglobin into the macrophages, or actually the macrophages swallow the red blood cells, whether they're in the liver and spleen, and the heme gets broken down to bilirubin, and all of that bilirubin winds up in the liver so that it can be secreted into the bile. So follow the common bile duct down to the end, and it joins with the main pancreatic duct to dump into the duodenum. And here's a close-up of that. So you see the common bile duct and the main pancreatic duct joining together to form the hepatopancreatic ampulla or duodenal ampulla. It's got a sphincter to make sure fluid doesn't flow backwards, and it projects out through the duodenal papilla. And all of that is going to be controlled by our hormones. So here we see the hormone CCK being secreted at the lower left. when the duodenum has fat in it. Mostly fat, also some protein. CCK gets into the bloodstream and stimulates the contraction of the gallbladder. Okay? And contraction of the gallbladder ejects the bile into the duodenum. CCK will also stimulate from the liver and pancreas the secretion of bicarbonate. All right, so the liver has lots of functions: metabolic functions, hematological regulation, and bile production. So we're going to take all the blood from the gut and also from the spleen, just because it's there. All that blood is going to flow through the hepatic portal system into the liver. Remember that blood is not well oxygenated, but it is nutrient rich if you have food in your gut and you're actively absorbing nutrients. So the liver cells are going to extract that nutrient. If there are any toxins in the blood, whether it is alcohol that you consumed or fungal toxins from bread that went bad, whatever, anything that's toxic is going to get absorbed by those hepatocytes. And the hepatocytes are going to remove and store excess nutrients. So if there's a lot of glucose in the blood or a lot of amino acids in the blood, the liver gets first dibs on all of those. It either uses them or it stores them. Any iron is going to get stored there. A lot of the vitamins that you can store, vitamin A, vitamin D, are going to be stored there. So that's a lot of your absorptive functions. We're going to see in the next chapter about the metabolism, but a lot of your basic metabolism, carbohydrate, lipid, amino acid metabolism, removal of waste, storage of vitamins and minerals, and drug inactivation are all metabolic functions of the liver. So what that means, what that should mean, is that your blood supply to the liver is pretty darn high, and that's a good thing. So it's also, because it has all these big veins, a blood reservoir. We talked in the circulatory system about how blood it's not really stored in your veins, it's circulating, but you can squeeze those veins and push more of that blood into the circulation. So that's why it's a blood reservoir. Phagocytosis and antigen presentation, that's part of your immune system. Synthesis of plasma proteins, albumin, and all those other plasma proteins, with the exception of the gamma globulins, which are antibodies. Most of your plasma proteins are made in the liver, including the ones involved in clotting, which is fibrinogen and thrombin. Your liver removes hormones from circulation. If you had a surge of epinephrine, adrenaline, because you were stressed, your liver and kidneys will help remove those. It removes antibodies, and it removes and stores toxins. All right, bile. Bile has really three main components. The bile salts are the only ones directly involved in digestion, and even there, they don't really digest anything. They emulsify fats, and we'll come back and talk about that. But bile also has a high pH, so it helps neutralize the acidity from the stomach. And the bile pigments, remember, are just waste products. Bile salts are these little molecules that are cholesterol at one end and a little salt at the other end. So the cholesterol end loves lipids and the salt end loves water, and that's what emulsification allows. You don't really waste these things. You do recirculate them. So in the small intestine, at the end of the small intestine and ileum, you can actually reabsorb bile salts. They get back into the circulation, go through hepatic portal, go right back to the liver, and get resecreted again. Obviously, some of them are going to get lost with every pass. So you do continue to make them, but they do recirculate. The gallbladder is storing that bile, and remember that those bile salts are mostly cholesterol. So when you get concentration of the bile, then sometimes those bile salts can coalesce into gallstones, and those are actually just little lumps of mostly bile salt, which means a lot of them are cholesterol. All right, and then the cystic duct is the way we're going to dump it out. So the gallbladder releases only when CCK, that hormone from the intestine, is stimulated. And that, remember, was stimulated by fat in the diet. So actually, without CCK, you don't get as much bile secretion through the gallbladder. And so anything that the liver continues to produce just gets secreted out. And people who've had their gallbladder removed, it's not a problem for them. They should eat less fat because fat actually stimulates through another hormone. It stimulates the production of more bile salts. But if you don't have a gallbladder, you just store the bile in the bile ducts, the remaining bile ducts. All right, moving on to the small intestine. This is most of your digestion and almost all of your nutrient absorption. We have 3 segments, the duodenum, the jejunum, and the ileum. The duodenum is the shortest segment. It's about 10 inches long. And that's where the chyme from the stomach mixes with the digestive secretions from the pancreas and the liver. And remember, both liver and pancreas are giving us some bicarbonate, raising the pH. The liver is also producing the bile salts, whether it came actually from the liver or gallbladder, didn't really matter. And the pancreas is giving us some enzymes. So all that mixing occurs in the duodenum. You're also going to do a little bit of absorption here. Absorption of most calcium and iron occurs here because it's still fairly acidic, and those 2 minerals are absorbed better in an acid pH. Plus, that's where your transporters are. As you move into the jejunum, this is about 2.5 meters long. Most of the digestion is going to occur here, and so likewise most of the nutrient absorption is going to occur there. And by the time you get to the ileum, even though it's the longest part, you're just kind of finishing up everything. And it's going to end at the ileocecal valve, which is a sphincter that prevents backflow, because once you get into the cecum, you don't want fecal material going backwards into the intestine. So here's a diagram showing you that about 10 inches of the duodenum. The jejunum is mostly superior and more on the left, and the ileum is mostly inferior and down on the right. And here's a view of the ileocecal valve. You're looking in through the ascending colon at the top and the cecum at the bottom, and you can also see the appendix in that picture. Internally, the histology of the small intestine, mostly in the jejunum and ileum, not so much in the duodenum. Well, you have circular folds throughout. Circular folds are just kind of spiraling folds that help mix the chyme with the intestinal secretions. And then on the surface, microscopically, you have intestinal villi. And the intestinal villi are going to increase the surface area for absorption, but so do the circular folds. So between the circular folds and the intestinal villi, you've got probably about 400 times as much surface area as you would if the lining were just smooth. All right, in each villus, you have a lymph capillary called a lacteal. Lacteal is the lymphatic capillary, and we'll take a look at that picture in a minute. And then you also have intestinal glands or crypts. So looking at this diagram, what I want you to focus on first is the villi. The villi are finger-like projections up. Now look between 2 adjacent villi. It's labeled at the top, the intestinal glands. These are the crypts, and down in the base of the crypts are going to be, that's where new cells are generated, and that's where your hormone-secreting cells are generated. In the interior of each villus, let's see, I think we have another picture. Yeah, there's a deep picture of the crypt, and then here's a picture of the villus. You can see that lacteal, colored green, just for contrast. They're, of course, colorless in real life. And then you have a capillary network within each villus. Each villus independently has its own lacteal and its own capillary network. On the surface, you can see columnar epithelial cells and goblet cells, and deep to that, the lamina propria, and then you're down, let's go backwards, into the submucosa, the muscularis, and the serosa. All right, so the cells are the cells that produce hormones, and then the stem cells are just generating new cells. As they divide by mitosis, they push up. So as they push up, the oldest cells are at the very, very top of each villus. And when they are worn out and die, they just get squeezed out. They get popped out from the top. So at the very uppermost tip of the villus, you're going to have the oldest cells, and they're just going to get pushed out. Okay, the submucosa of the duodenum, specifically just the duodenum, has a very alkaline mucus secretion, and that protects the duodenum and helps to counteract the acidity of the stomach acid. And if that doesn't work right, if you get erosion of that mucus, then you get a duodenal ulcer, an ulcer due to the acid of the stomach. The intestinal juice is not really enzymatically rich. It is going to just help wet the chyme, suspend the food particles in the chyme. It's going to adjust the pH of the chyme, and it's going to keep everything mixed and moving. The enzymes of the small intestine are mostly like embedded in the intestinal microvilli. That is the epithelial cells of the intestinal microvilli, mostly at the tips, have these enzymes sticking out. We call them brush border enzymes. And chief among these is one called enteropeptidase. That's the one that activates trypsinogen. Remember, trypsinogen came from the pancreas. And so it got secreted through the pancreatic ducts and into the duodenal papilla before it even ever got activated. And that's good because it keeps your pancreas from digesting itself. In fact, if you have inflammation here, pancreatitis, you can actually auto-digest the pancreas. This is pretty common in dogs that have been fed food that's too rich. Don't feed your dogs barbecue. And we'll come back to the other brush border enzymes. In terms of the muscular activity, once the chyme arrives into the duodenum, you're going to have some peristalsis moving it slowly to the jejunum, but you need to have time to get it mixed with those bile and pancreatic secretions to raise the pH and protect the jejunum from that acid, and also to absorb the calcium and the iron. These are local control, not so much central nervous system. But there are some central nervous system reflexes, the gastroenteric reflex and the gastroileal reflex. These start in the stomach. That's what gastro tells you. So anytime you have food in the stomach or digestive activity in the stomach, it actually is going to stimulate the intestine and the ileum, and it's just going to move stuff along faster. So you are actually speeding up the processing of food when you eat again. Every time you eat again, you speed up the motility of the rest of the intestine. All right, so here you see the gastric reflexes, the gastroenteric reflex, and the gastroileal reflex. Later on, we'll see there's a gastrocolic reflex affecting the colon. The neural and hormonal mechanisms that regulate the small intestine are coordinated with the gastric regulation and the cephalic regulation, and they're all going to activate digestive secretions when there is food in the duodenum. So our neural mechanisms include parasympathetic innervation. We're going to inhibit through sympathetic only if there's something nasty in your stomach or you otherwise got turned off appetite-wise. And we're going to secrete not just neurotransmitters, we're also going to have some hormones secreted. So here's this list of hormones, some of which we've already seen. Gastrin is secreted by the duodenum when it's ready for more. So gastrin gets secreted when the pH of the duodenum rises back up a little bit, because that means it's ready for more acid. Secretin, GIP, and CCK all inhibit gastrin and gastric motility because they're secreted when acidic chyme just moved in from the stomach, and that's going to tell the stomach just to wait. Secretin does a couple of other things I haven't mentioned yet. Secretin stimulates the liver to produce more bile, and secretin also stimulates the pancreas to secrete more bicarbonate. GIP just inhibits the stomach as far as I know, but cholecystokinin, we've only mentioned one thing it does so far, and that is to contract the gallbladder, but CCK also stimulates the secretion of enzymes from the pancreas. VIP is going to cause dilation of the blood vessels. I think I have all these things listed further down. Yeah, and enterocrinin is going to help stimulate mucus production. So that's just sort of a big overview of what those hormones are doing. And here's a diagram showing you. So I'm going to just direct you to the book to kind of go through the diagram. Pay attention to whether something's stimulating or inhibiting. All right, absorption in the small intestine. We haven't even talked about digestion yet, but we're going to come back and talk about digestion separately. And here we're just kind of looking at the big picture. So we're going to absorb most of our organic nutrients, a lot of our water and ions, just a lot of things are going to be absorbed in the small intestine. And then we'll move on to the large intestine. And the large intestine is divided into regions, the cecum, the colon, and the rectum. And the cecum is sort of a little blind pouch that just receives the stuff coming from the ileum through the ileocecal valve. And at its inferior end is the appendix. It's called vermiform. Vermiform just means worm-shaped. And the appendix is actually part of your immune system. It's dominated by lymphoid nodules, and it's like an alert system. If there are bacteria that don't belong to you, or if those bacteria are trying to invade the ileum, that would be bad. So we all know you can live without your appendix, but the appendix is part of your immune system. The colon is the biggest part. The colon is divided into 4 regions, and each region has sort of this appearance of this pouchiness. The pouches are called haustra, and they're held in place by longitudinal bands of smooth muscle called the tinea coli. And when they contract, they create these pouches called the haustra. And then you have these omental or epiploic appendages, which are just little sacks of fat that we're not really worried about. So here are the 4 regions of the colon: ascending, transverse, descending, and sigmoid. And the ascending is going to start right at the cecum. So food goes, or not, it's not food anymore, it's fecal material. It goes up the ascending colon. It turns right at the colic flexure. Then it enters the transverse colon, which goes across from right to left. That turns down at the left colic flexure into the descending colon. And the descending and ascending colons are both retroperitoneal. The transverse colon and sigmoid colon are not retroperitoneal. As fecal material moves into the sigmoid colon, that's what empties into the rectum. So the blood supply to the large intestine is mostly from the mesenteric arteries, superior and inferior, and then drainage through the corresponding veins. So here's your diagram showing you all those parts, and by now I'm just pretty sure you've learned them in lab. Last part's the rectum. Rectum refers to rectus, which just means straight. Let's back up and look, and here you can see the rectum is just sort of this vertical midline straight portion of the colon, or of the large intestine. And the rectum can store feces, and then it can expel feces. The anal canal is just that last little part. The anus is the opening of it. And the anal canal is a stratified squamous epithelium. The anus itself is a continuation of the skin. And it has 2 sphincters. An internal sphincter is smooth muscle, and the external sphincter is skeletal muscle. And we'll see the same thing with the urinary system. So here you can see those sphincters. And the external sphincter is under voluntary control. So this is when you learn how to control your bowels, evacuation of the bowels, you're learning to control the external sphincter. Histologically, when you look at the lining of the large intestine, lots of goblet cells. Remember, they produce mucus, so that's for lubrication and protection. Yes, you have intestinal glands, but these are just full of goblet cells, so they're not secreting any digestive enzymes. They may be secreting a little bit of fluid, but mostly the job of the large intestine is to absorb fluid. And then lots of lymphoid structures for your immune system. So here you can see those intestinal glands in the large intestine just full of goblet cells. And there's a histological view. So functionally, the main job is to absorb. And it says reabsorption because a lot of this is from the fluids you secreted. You secreted about 1 liter of saliva, about 1 liter of intestinal fluid. So you have to reabsorb all that stuff. You're going to recirculate your bile salts here. Some of what you absorb is produced by bacteria. That includes some of the B vitamins and also vitamin K. But if bacteria are producing toxins, well, you can absorb those as well. So after you've absorbed everything you want, you have compaction into feces and then storage prior to defecation. The microbiome is all of the microbes, bacteria mostly, some fungi, some viruses, And the ones that are in the intestine are called coliform bacteria. These are things like E. coli. But there's lots of different coliform bacteria. Most of the time, they're not just harmless, they're actually helpful. The bacteria produce some of the vitamins, some of the B vitamins, and vitamin K. So the B vitamins are biotin and vitamin B5, and so you really don't need to ingest these necessarily, but notice that vitamin K is used in clotting. I think we should have already known that. And the B vitamins are involved in different aspects of metabolism. The organic wastes include the bilirubin, which remember was the waste product of hemoglobin. And the bacteria actually get some nutritional value out of that, and they take that green bilirubin and convert it to the brown pigments we associate with feces. If you have someone who's not secreting bile properly, then their feces can be just like the palest tan color from lack of bilirubin. All right, so bacteria are also feeding on anything else that you have left for them. Some of what they produce are called indole and scatole. These are the ones that are going to give feces their bad rap in terms of smell, that along with hydrogen sulfide. And the carbohydrates we eat that we can't digest, they just love them. And a lot of those are going to provide gaseous end products. We had our gastroileal reflex already covered, the gastroenteric reflex already covered. There's also going to be a gastrocolic reflex. All of that is going to propel faster movement of things into the cecum or into the colon. It's going to increase the motility of the colon. It's going to give us what we call mass peristalsis into the rectum, and that eventually is going to lead to a defecation reflex. So the gastrocolic reflex causes people to have to get up and go right after eating a meal. This is why usually if you fed a baby, you have to change their diaper after they eat. All right, so we have some positive feedback loops. We have those short reflexes. Remember, those are just managed locally through local nerve reflexes. And they're going to just keep peristalsis going. And then parasympathetic, remember, is a long reflex because it's coordinated through the central nervous system. In the sacral region of the spinal cord, you have parasympathetic neurons that are receiving stretch impulses from the rectum, and that's going to stimulate defecation. So our defecation reflex is going to be regulated by the movement of material into the rectum, and then you've got to relax the sphincters. Intrinsically, babies do this without having any control over it, but your somatic nervous system can be entrained to control the external sphincters so that you can resist the urge to go if it's not convenient. And those are carried by the pudendal nerves, which are going to supply the nerves at the pelvic floor. So there's a diagram showing you the control of the internal and external sphincters. Okay, last topic is chemical digestion, getting our nutrients digested. So the main nutrients we digest are the carbohydrates, the lipids, and the proteins. Vitamins are not digested. We have to absorb them the way they are. Minerals you can't digest, and of course water. So we're going to do physical breakdown. We're going to do this through mechanical digestion, which includes chewing, but also that mixing activity. And then we're going to do molecular breakdown. And then we have to absorb them into the bloodstream, and we got to get them where they need to go. So the main process of chemical digestion is going to be through the enzymes. And we're going to have enzymes to break down some of the carbohydrates, most of the proteins, some of the lipids, and the nucleic acids. And so we have carbohydrases to break down carbohydrates, proteases, and lipases, obviously. So our digestive enzymes are secreted, as we've already seen, mostly by salivary glands, stomach, and pancreas, and then the brush border enzymes in the small intestine. So starting with carbohydrate digestion, you cannot digest them all. You can digest starch, and you can digest glycogen, and the amylase that comes from your salivary glands and your pancreas can break those down all the way to maltose, which is a disaccharide. You also ingest sucrose, that's just table sugar. Lactose is milk sugar. And so step 2, the brush border enzymes are specific for those 3 sugars. And what they're going to break it down to is monosaccharides, predominantly glucose, but also fructose and another one. But I do also want to point out that there are some carbohydrates you eat that you cannot digest, and they go through your entire digestive system undigested until they hit those bacteria in the colon, and they're feeding the colon bacteria. Also, milk contains some small saccharides that feed those bacteria as well. Lactose is interesting because lactose is milk sugar. Mammals evolved to feed milk to babies. So the fact that human adults can still tolerate milk sugar is due to a mutation. So if you are lactose intolerant, you're the normal, default, non-mutated human type. If you can still drink milk as an adult, that's the result of a mutation that allows consumption of milk. Once we have our saccharides, our carbohydrates, down to the monosaccharide stage, we can absorb them. And we're going to absorb them by a combination of processes depending on which actual monosaccharide it is. So in this figure, we're going to see just sort of we'll do the same for lipids and proteins. In the mouth, some salivary amylase. In the stomach, that can get digested down to the disaccharide maltose or some trisaccharides, but you're not going to finish it until you get to the small intestine where you add the pancreatic amylase. And then finally at the intestinal mucosa are brush border enzymes, lactase, maltase, and sucrase. We absorb them by either facilitated diffusion or by co-transport, and then they pass through the epithelial lining to get to the bloodstream, and they're going to get carried through the capillaries in the intestinal villi. And from those capillaries, they're in the blood, so they go straight to the liver through the hepatic portal circulation. Okay? Lipids. This is a little more complicated because lipids don't mix with water very well. So one of the first things we have to do is mix the lipids with the bile salts. Remember, those were cholesterol at one end and ions at the other end. And they form these little blobs we call micelles. And that gives the lipase from the pancreas access to the triglycerides, which get broken down to monoglycerides and fatty acids. Now, that's not the only lipids. There are steroids, there are phospholipids, there are lipid-soluble vitamins, and all of that is in a micelle. And as those micelles reach the surface of the epithelium, the lipids are just going to diffuse into the cell. Let's watch what happens then. So a little bit of lingual lipase in the mouth, up in the oral cavity, not significant. Most of the action's in the small intestine, but it's due to the bile salts and the pancreatic lipase. Look at the intestinal mucosal cell. We're going to take our monoglycerides and fatty acids, build them back into triglycerides. We're going to combine them. Let's go back to the previous slide. Combine them with steroids, phospholipids, vitamins, and proteins. We're going to form a lipoprotein called a chylomicron. Chylomicrons are big. They're too big to pass through the membrane, so you secrete them by exocytosis, and they're too big to go into the capillary, so they go into the lacteals, those lymph capillaries in the intestinal villi. And so the chylomicrons are loaded into the lacteals. Well, from there, they go through the lymphatic system of the mesentery. They get into the thoracic duct, which is the main opening into into the cisterna chyli, opening into the thoracic duct. Remember that the thoracic duct passes above the diaphragm, left of the aorta, makes a left turn, and dumps into the left subclavian vein. Now, this is really weird because what we're doing is we're taking all this milky chylomicron-rich lymph and dumping it straight into the vein, the subclavian vein on the left side. And from there, it's going to drain into the right side of the heart through the superior vena cava. And from there, it's going to go out to the lungs and then back to the left side and out through the aorta. So we're just dumping these chylomicrons into the general circulation. Eventually, they'll get back to the liver, and the liver will deal with them. Okay, proteins. We're going to break our proteins down through all of those proteolytic enzymes, starting with pepsin in the stomach, trypsin, chymotrypsin, and carboxypeptidase in the pancreas, or from the pancreas, but in the small intestine. When we get to the level of dipeptides, we've chopped them down to dipeptides. We have dipeptidases, which can break them down into individual amino acids, and now we're ready to absorb them. So starting with pepsin in the stomach, polypeptides through trypsin, chymotrypsin, carboxypeptidase, and then to the brush border dipeptidases. We take them in a lot like we did with the sugars, facilitated diffusion, co-transport, and again, like sugars, they're going to go into the capillary, and that's going to carry those amino acids straight to the liver through the hepatic portal circulation. Okay, nucleic acids, very briefly. You've got some enzymes called nucleases, which break down the nucleotides, the DNAs and RNAs. We're going to absorb those by active transport and be done with them. Water, you're not digesting it, but you are absorbing it. So let's take a look. Water is taken in from what you ingested, right? About 2 liters a day. Saliva, about 1.5 liters. Gastric secretions, another 1.5 liters. And then intestinal secretions, about 2 liters. And colon secretions, very small, only about 200 milliliters. But look at the absorption. 8 liters a day absorbed in the small intestine, another little over 1 liter absorbed in the colon, and only about 150 mils lost in the feces. But that absorption in the colon is not that straightforward. So you have to absorb ions in the colon, actually in the small intestine as well, but that creates an osmotic gradient that then allows water to be absorbed. So water absorption, remember, strictly by osmosis. It's controlled by the absorption of other solutes, all those organic solutes in the small intestine, and in the large intestine, it's a lot of bicarbonate and other ions, including sodium. And calcium absorption is, again, that's mostly in the duodenum. It's regulated by the hormone calcitriol. Hey, we already studied these hormones. So if we back up one slide, aldosterone absorption is regulated by, or aldosterone regulates sodium absorption and potassium secretion. So refer back to those hormones. You've already studied if you need to, but there are other ions, magnesium and iron and other cations, and you have to have some regulation of that, and the anions as well. But in general, you're just going to absorb the minerals you need, the ions you need. Our vitamins, we subdivide into water-soluble and fat-soluble. B's and C are water-soluble. A, D, E, and K are lipid-soluble or fat-soluble. The fat-soluble guys are going to go into the micelles and then into the chylomicrons with lipid. And the water-soluble guys are just going to dissolve in the bloodstream, no problem. All right. Just a few words on aging. Just remember, in general, when you get old, everything kind of slows down. You lose muscle tone. Your risk of cancer increases simply because of the cumulative random mutations to DNA. And the elderly some old people tend to be fixed in what they eat. If they eat the same thing for breakfast, lunch, and dinner every day, they may be just not well nourished because they don't have enough variety in their diet. So that's something that has to be watched. And then finally, you've got your integration with other systems, which I'm hoping y'all are always at least going through. It's a good way to review for the final exam and also give you just a more holistic view of how the body works as a whole. Well, I believe that finishes our discussion of the digestive system, and I hope that if you have any questions, you'll contact me and let me know.